I want us to talk about immunobiotechnology by looking at immunotechniques or techniques that utilize immunoreactivities. I will start off with an introduction of some basics concepts, such as what are antigen antibody reactions, what is affinity, what is avidity, what is cross-reactivity, among others. So antigen antibody reactions, usually we say that an antigen is a substance or molecule that once you introduce it within a body, it is going to trigger the production of antibodies through the immune system, which in turn will now neutralize or kill that particular antigen because it is normally recognized as foreign and may be potentially harmful. So these invaders, for example, in health, human health, could be things like pollen, could be cells such as bacteria, viruses, etc. So antigen antibody reactions are very important when it comes to immune reactions, because with this we are able to eliminate any foreign pathogens that may be invading the human body, the animal bodies, including also microorganisms. They also have a fighting response against any foreign antigens. So some characteristics of antigens is that they can be proteins, they can also be in form of polysaccharides or carbohydrate moieties, lipopolysaccharides, which have lipids and carbohydrate moieties, nucleoproteins, like nucleic material or histones that normally uh, bind to the DNA. We have glycoproteins. They could also be in form of steroid hormones, as bacterial cells and viruses. They can also be made synthetically, that is like synthetic polypeptides and polymers. Now, some important words you will come across is immunogenicity, which is a property of now the antigen, and antigens can also be called immunogens if they are able to induce a detectable immune response. Then antigen specificity is now the property where an antigen or part of it, because it could be maybe a surface protein of that antigen, is able to react with a specific antibody. Then antigenicity is given by a surface structure of now the immunogen, so that now within the antigen we have different surface proteins, surface moieties that could be polysaccharides, lipids, like we have seen. So these different parts of the structure of an immunogen or antigen, we call them antigenic determinants. And normally now the organism, let's say the human being, is going to respond to either one or more of those antigenic determinants. So the number of antigenic determinants usually will vary with size and also the chemical complexity of the macromolecule. So if the macromolecule, for example, we have egg oval albumin, which is about has a molecular weight of 42,000 Daltons, then it has about 5 antigenic determinants. Then thyroglobulin, which has 700,000, then has about 40, because it's definitely a bigger and more complex macromolecule. The antibodies, on the other hand, usually will have the structure as you can see in this diagram here, usually like a Y-shaped structure, which has what we call a heavy chain. In the big middle here, it's orange in color, and then we have light chains on the outside. And we have the binding sites for the antigen, which is called the antigen binding site on these ends. Now we have different types of antibodies. We have IgG, which is monovalent and normally highest opsonization and neutralization activities within the body. and it can be grouped into some subclasses, that is IgG1, IgG2, IgG3, IgG4. And then we have IgM, which is pentavalent. Yeah, it has 5 moieties, and it is normally produced first upon antigen invasion. Yeah. Then we have IgA. IgA can conform into different structures: monovalent, divalent, trivalent. Then it is expressed mainly in mucosal tissues. That's why you'll find it along the GI tract, like here. And then we have IgD, which is monovalent. Its function is not very well known. Then we have IgG, monovalent, which is mainly involved in allergies. Yeah. Then we do have a characteristic of this antigen now binding to antibodies, which we call one of them being the lock and key concept. And this I'm sure you have seen also when you have enzyme and substrate reactions. So the lock and key concept is that the combining of an antibody is located on the FAB portion of the molecule and is constructed from hypervariable regions of the heavy and light chains. So we normally have it a kind of lock and key. So they combine under lock and key concept, and the bond that binds the antibody to the antigen is non-covalent. Yeah. That means it could be bonds like hydrogen bonds, electrostatic bonds, van der Waals, and also hydrophobic bonds. That means because it is non-covalent, then it's reversible. So these antigen-antibody reactions usually can be reversed. Yeah. Now we have some other two concepts, that is what we call affinity and avidity. So we often call binding partners ligands or receptors, yeah, and they have one or more binding sites for their partner. So what happens, for example, in this case, when we have a monovalent binder which has a single binding site, it means that only one part of your molecule is going to bind to the receptor. And here, the binding strength of these two will depend on what we call affinity. That is how well each site binds independently. So how this binds to this, how this is going to bind to another receptor, etc. So the binding strength is often reported as the dissociation constant. Remember, we said that this binding is reversible. So the dissociation constant, KD, so the higher the affinity, meaning the more tightly they bind, then the lower the dissociation constant. Now, when we have multivalent binders, meaning that we have ligands that are binding to receptors more than one, we could have it being bivalent, trivalent, etc. So the binding strength here depends on avidity, because one binding will affect the binding of the other because they are working together here. So the effects that come with multiple proteins working together, normally it will be easier for one to bind if another one has already bound. Yeah, so that is what we call multi-valent binders. So in more closer look of these two terms, so affinity is the strength with which an antigen and an antibody molecule interact with each other at that particular binding site. And it usually considers the interaction between one single epitope and a paratopia. So when we have low affinity, it indicates that we have a weak interaction between our binding molecule, and therefore it is easy to dissociate that particular complex. If we have a high affinity, then it indicates that then we have a very strong bond, and for us to break that bond, it will take longer. So high affinity antibodies bind to a larger number of antigens in a shorter duration of time because they're able to attract us quickly. So the binding between antigen and antibody molecule, we already said, is a reversible reaction. That's why we have antigen antibodies to antigen antibody complex can be reversed. Now, avidity, on the other hand, we said is a measure of the overall strength of binding of one antigen which has several or many antigenic determinants to multivalent. antibodies. So affinity refers to know the strength of a single antigenic determinant with one antibody, like you can see in this diagram here, one antibody, one antigenic determinant. But now avidity will mean multiple. For example, this avidity for one antibody which has two binding sites binding to two different antigenic determinants. So usually the avidity is influenced by both the valence of the antibody and the valence of the antigen. That is to mean that valence of the antibody means, remember we said monovalent, which is one single antibody, but we have those that are like pentavalent. So that will also affect the avidity or the strength of binding. Also, the antigen, how many of them are there? Is it two? Is it three? Is it four? Yeah. So for example, here we have affinity of 10:4. Here we have avidity of 10:6. Here we have avidity of 10 to power 10. So avidity is more than the sum of the individual affinities. So the measure, in summary, of an avidity is the overall strength of binding of an antigen with many antigenic determinants as well as multiple antibodies. Yeah. Then this is influenced by both the valence of the antibody and the valence of the antigen, more than the sum of the individual affinities. Yes. So affinity is now finally the strength of binding between a single antigenic determinant and an individual antibody combining site. And avidity is the overall when we are binding between multivalent antigens and multivalent antibodies. Now, it is important when we talk about specificity because this also comes into play when we are looking at disease or even laboratory tests that utilize this immune based in reaction, because specificity refers to the ability of an individual antibody combining site, yeah, to react with only one antigenic determinant. That if it binds to, if you're having these several, the purple, red, green, yellow, blue, and or orange, that this particular antibody, this antigen binding site will only bind to one, like the yellow one, and not the rest, even if they are sharing shape, yeah. So that is very important so that each antibody binds to a specific antigen. So ideally, that is what happens, or what we normally have in our mind is one antibody should only bind to one protein or antigen. And that's why we do a lot of immunoblotting, Western blotting, etc. But in reality, we have a challenge of what we call cross-reactivity of antibodies, that sometimes one antibody will bind to two or more antigens, yeah? And this usually can be as a result of certain things like maybe we have, if this was antigen A and this was antibody A, you can see the structure here, but you can have a shared epitope with another antigen B here, which looks alike as the antigen A. So the antigenic determinant, this surface macromolecule, this surface antigenic determinant is similar to the antigen A. So that means that anti-A antibody is also going to bind to the antigen B. Yes, we can also have similar epitope so that now they are similar. So this is another antigen C, and the design is the same, although not exactly the same. You can see that the design, so that the lock and key concept is still able to work in this case. So cross reactions arise because the cross reacting antigens either are sharing an epitope in with the immunizing antigen, or because it has an epitope which is structurally similar to one of the immunizing antigen, what we call multispecificity. So usually in the lab, how we go about this is by having at least another maybe secondary antibody, so that you can increase the number of antibodies that are detecting your antigen, and that way you're able to at least somehow eliminate. And that's why sometimes also when you start to do research, and you want to maybe develop a diagnostic kit, you have to look for maybe organisms or proteins that may be in the same family. Let's say if it is coronaviruses like SARS-CoV-2 and other coronaviruses, you'd want to understand, do the antibodies you're having against SARS-CoV-2 also react with other coronaviruses apart from or in addition to the SARS-CoV-2, so that at least you can be sure that you're actually detecting the correct viral antigen. So that is okay for now. I want us to go to the next one.